students to this module of e-learning. I am Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present to you this module to the first year students of Botany, Semester 2, BOC 102, Biodiversity 2, Vascular Plants. The module name, Taxonomic Evidances from Cytology, belongs to Unit 5, Taxonomic Evidances from Palynology, Cytology, Photochemistry, and Molecular Data. The outline of the module will be, students, definition of cytology, the basic evidentiary characters of chromosomes, and the taxonomic evidence from cytology. Learning outcomes. Once the student completes this module, the student will be able to explain the terms taxonomic characters and cytology, describe the evidentiary characters of chromosomes, and explain the taxonomic evidences based on cytology. Now, students, what is the meaning of cytology? It is the study of the morphology and physiology of cells. Normally, anatomists deal with the shape, size, wall, pattern, etc. But cytologists deal with the internal organelles of the cell and detailed structure of the cell wall. Now, the chromosomes in the cell are Chromosomes are the carriers of genetic information, and they have considerable significance in evolutionary studies. Taxonomy in relation to cytology. The application of cytological data in elucidation of taxonomic problems. Now, there are various attributes, students, the cytological characters, of the chromosomes, like chromosome number, morphology, size, behavior during meiosis, aberrations in reproduction, and these are all important and used in taxonomic studies. They provide an idea of genetic relationship between plant populations. Now, where the cytological evidences are used? They are used to distinguish taxa, to determine the origin of groups, to understand evolutionary history of related taxa, particularly those at infraspecific and specific levels. In addition, the homologies of chromosomes in hybrids as determined in meiosis is significant indicator in knowing degree of genetic relationships. There are a large number of workers, students like Darlington, Janki, Darlington and Wiley, Love, worked a lot on this chromosome number of various plants, and the International Association of Plant Taxonomy published an index to plant chromosome number in a series of Rhagnum vegetabile in 9 volumes. Now, based on chromosome number, students, we will see what evidences are there. The chromosome number is constant and same in all individuals and species, and it is used as a confirmative property to distinguish one species or the species from members to other species. Chromosome counts are reported as diploid, that is 2n, and haploid as n. In angiosperm, if you see, the chromosome number varies greatly, that is n is equal to 2 in Haplopappus gracilis, member of Asteraceae, and highest n is equal to 132 in Poa litorla in Poaceae. Now, if you see, most of the angiosperm have a chromosome number ranging between n is equal to 7 and n is equal to 12. Specifically, it is a characteristics of Embolrelaceae that it is 2n is equal to 26 and 2n is 16 of Trimeniaceae. If you see the separation at the family level, we see that the traditional way of segregating the Ranunculaceae family, they have placed it in 2 tribes, Heliboriae and Anemoniae and Clematidae. Now, in Heliboriae, these are the genera which have few carpels, which contain more than 1 seed per carpel, whereas in Anemoniae and Clematidae, those genera are the ones which have many carpels and which are one-seeded and indehiscent students. However, the family has been conveniently divided into two tribes by means of the karyotype morphology. Now, two groups have been made. One group of genera is dominated by basic chromosome number X is equal to 7, whereas the second group by X is equal to 8. Now, the X is equal to 7 group, they have small chromosome number with conspicuous heterochromatic chromocenters, whereas the second group with X is equal to 8, they have large chromosomes and are irregularly distributed heterochromatic regions. At the generic level, how the separation is done? There is Cicenda filiformis and Microcala pusilla. Now, they are placed in Cicenda genus. Now, they are separated based on chromosome number. Cicendas filiformis, n is equal to 13, and Microcala pusilla, n is equal to 10. In another genus, that is Staphyrosia, all the species of this genus in Fabaceae family have 2n is equal to 22, except there is one species which is Staphyrosia constricta that has 2n is equal to 16. So now, this Staphyrosia constricta has later is raised to the level of the genus Sphinctospermum. In the tribe Boraniae of Rutaceae, the chromosomal base number varies from 7 to 19. So the members are rearranged to have a unique system. Now students, have a look at these two pictures. One is Monotropa hippopoetis and Monotropa hypophagia. Now these two different species, they look alike morphologically. But now they are separated based on chromosome numbers. One is hexaploid showing 2n is equal to 48, whereas hypophagia is a diploid showing 2n is equal to 16. Some species generally show a single chromosome number. Certain population of infraspecific taxa may sometimes show a different chromosome number or even a different chromosome morphology. And such populations or infraspecific taxa constitute the cytotypes. Now let's see an example of a cytotype, how the separation has been taken place there. Now there are two cytotypes of the species Veronia prostrata. One is Veronia prostrata, first species, and the second. Now, based on the chromosome number, these two cytotypes are raised to a subspecies level, wherein one is raised to this subspecies, that is prostrata, showing n is equal to 8, whereas the other one is raised to Shirae, showing n is equal to 16. So, in case of aneuploids, these are plants with basic number of chromosome with some addition and deletion of a few chromosomes. Now, we see the duplication of chromosome number leading to polyploidy may prove to be of taxonomic significance. For example, in Datura, we have monosomics, that is 2n is equal to 1. In Triticum, we have nullisomics, 2n is equal to 2. In wheat, we have 2n is equal to 2n plus 1. There are trisomics. And in some plants, we have tetrasomics, which have 2n is equal to 2. So, this is based on aneuploidy. Whereas, based on morphology, chromosome morphology, that's a structure. A study of chromosome morphology is very informative to the taxonomists in assessing affinities and modes of origin of separate species. Now, there are a large number of characters like the shape of the metaphase chromosome, the position of the centromere, the occurrence and position of secondary constriction, which demarcates a satellite is important in chromosomal identification and characterization. Based on chromosome morphology, they show a lot of variation depending upon the position of the centromere and the secondary constriction. Now we have metacentric, submetacentric, acrocentric, telocentric types. Now, Babcock in the year 1947 separated closely related genera on the basis of chromosome number and morphology. What Babcock did is, Youngia is separated, now that this is Youngia students, is separated from Crepsis, this plant, while Pleurothecia was merged with Crepsis. Now this important work of Babcock and Crepis shows how chromosome morphology coupled with chromosome number is of considerable importance in the genetic and taxonomic phases of study. In genera like Crepis and Plantago, the large size and small number of their chromosome have been of great value in this type of study. Hutchinson also did some work. He separated Pandanus typhus sporogonium on the basis of chromosome morphology and kept them under two different orders, Pandanales and Typhales. Similarly, Cyperaceae and Uncasey were separated due to distinct floral structures, and they both show holocentric chromosomes and are now closely related. Now students, karyotype of plant is very useful for the classification in some plants. Now what is this karyotype? It's an analysis of the chromosomes, how the chromosomes are arranged. A large number of studies have led to the conclusion that symmetrical karyotype is primitive and asymmetric karyotype is more advanced. Due to the different karyotype in this plant, Butomus, it is separated from Limnocaris and is kept in Butomaceae, while others are retained in Alistomacea. Another example, students, is of agave and yucca. You can see the photographs here. Agave species, because it possesses an inferior ovary, it is placed in Amaryllidaceae, whereas yucca shows superior ovary and it's placed in Liliaceae. But now, because Hutchinson shifted both this genera to Agaveaceae on basis of overall similarity, bimodal karyotype is seen in Agaveaceae. The members of Agaveaceae shows two types of karyotypes consisting of 5 large and 25 small chromosomes. Now, the karyotype study of the members Agaveaceae confirms shifting of agave from Amaryllidaceae and yucca from Liliaceae into Agaveaceae. The family Agaveaceae contains about 16 genera such as agave and yucca. Now this shifting was basically done because both the plants show a distinctive bimodal karyotype. Based on chromosomal behavior, students, it provides clues about the cause of sterility and fertility among populations. Structural differences in the parental chromosome is the main cause of the sterility. If the two sets of parental chromosomes are homologous, the plant seems to be fertile. Meiotic behavior of chromosomes is helpful in comparing the genomes to detect degree of homology. Now, example, Triticum aestivum is a hexaploid, where A is derived from Monococcum species, whereas B from Aegilops peltoides, and D is derived from Aegilops squarosa, which is diploid. It has already been discovered, students, that evolutionary development involves, in addition to alterations in chromosome number, their size, changes, and structure. So that analysis of this cytological characters may also shed important light on species relationships. Observations of many spermaceae have shown that this aspect of cytology is sometimes valuable in taxonomic discussion. So students, you have seen how there are various examples have shown that how there are various evidences from the cytological point of view with the chromosome structure, number, behavior, how they have helped to solve the disputes and bring some families together or place them far apart. These are the references, students. And these are the online references. And thank you, students, for a patient hearing.